Welcome to the cutting edge of RNA therapeutics. I'm your host, John Cook. I'm professor and chair of the Department of Cardiovascular Sciences and also the director of our Center for RNA Therapeutics here at Houston Methodist Hospital. Joining me in the studio is Dr. Christopher Brannon, who's going to introduce our visiting professor and will also be introducing our discussants.
Yep. Thanks, John. It's my pleasure today to welcome Dr. Michelle Hastings, who is the Pfizer Upjohn Research Professor of Pharmacology at the University of Michigan Medical School and the director of the Center for RNA Biomedicine's mRNA Therapeutics Initiative there. Before joining the University of Michigan, Michelle was professor and director of the Center for Genetic Diseases at the Chicago Medical School. And Dr. Hastings is a National Academy of Sciences Kavli Fellow and an inaugural board member of the Society for RNA Therapeutics. She holds numerous patents and is on the advisory board of numerous companies, and she's really a pioneer in the field of RNA therapeutics. Her group is focused on the genetic basis of disease, and they've developed some antisense oligonucleotide technologies that can modulate pre-mRNA splicing. And they've published a lot of amazing work showing that those technologies can be effective at correcting gene expression in diseases such as Usher syndrome, Batten disease, cystic fibrosis, and other diseases. So we're real excited to have her here to tell us about some of her work developing these therapeutics. We're also joined by a discussion panel that includes Dr. Vrtat Shah, who's a research associate in my lab, Dr. Ricardo Noriega, who's a postdoctoral fellow in my lab, and Dr. Dan Kiss, who's an associate professor here at the Center for RNA Therapeutics at Houston Methodist Research Institute.
Well, welcome everyone, and particularly Professor Hastings. Thank you for joining us, Michelle.
Well, thank you for having me. I'm delighted to be here.
You just moved fairly recently to University of Michigan and to initiate the RNA therapeutics program there. Tell us a little bit about that.
Yeah, I just moved here in May, actually, from Chicago, and I'm really excited. The University of Michigan made a strong statement that they want to bring RNA therapeutics to the university and to their patients here at the medical school, and broader than that, and so we started the RNA Therapeutics Initiative with really intending to be sort of a one-stop shop for people that want to get started or bring forward their discoveries in their lab, as well as to patients that have diagnoses with diseases that can be treated with this type of therapy.
Mm-hmm. And Michelle, when you say a one-stop shop for RNA therapeutics, is that referred to going all the way from the bench to the bedside?
That's our vision. In some cases, we think it's possible, and there's really demonstrations that this is happening from really, literally, we've been talking about bench to bedside for many years, but literally from the bench to the bedside, discoveries that are made. We can help researchers design the best type of an approach using an RNA therapeutic, help them understand the experiments that need to be done, how to begin that development at pre-IND. We have the resources here to also help with partnering or further development on the resources for that. So we really think the medical system here, the hospital, has access to the patients as well as the doctors, physicians that can help us with clinical trials. And so we're really excited about really the breadth and the ability to facilitate this kind of process. Really,
really exciting. Well, with that said, why don't we launch into your talk on Spline-Switching antisense oligonucleotides for the treatment of disease. And Michelle's been kind enough to say that as questions come up during her talk, the discussants will be allowed to ask those questions, and anyone calling in can as well. So, Michelle, please.
All right, thank you. Again, I'm so excited to be here. Just some of my disclosures. I just want to really briefly introduce my lab because they're really the most important feature of what we do. And again, we just moved to the medical school, Department of Pharmacology at the university here. We're really an RNA biology lab, so we work on RNA, looking at basic mechanism of RNA As I mentioned in the opening here, we're really interested in developing therapeutics based on understanding the mechanism of gene expression in RNA expression. We're pretty agnostic in terms of the disease. We work on a lot of neurodegenerative diseases, but also on other genetic diseases like cystic fibrosis and Usher syndrome. And we love to collaborate through either partnerships with other academicians or consulting companies, licensing and developing. And so really what I was doing at sort of a mini scale in my lab, I'm really excited to talk about this with other people and work with other people to facilitate this at a grander scale throughout our labs. And so I work at Michigan with the Center for RNA Biomedicine, which is headed by Mats Lungman and Nils Walter, who are the two directors. And they brought me in to have this mRNA therapeutics initiative at the university, as we just mentioned. And I'm working with the new clinical director, Peter Todd. And again, the vision is to have a real true bench to bedside where we're translating our discoveries at the bench, but also bringing in the patients. And so we envision sort of a scheme like this, where we will be discovering things in the lab, designing oligonucleotides, manufacturing them, and getting personalized medicine into patients, but also diagnosing patients with a focus on genetics and genomics, and then using bioinformatics and testing here that we have expertise at Michigan, and going back to the lab and going through this process in the other direction. And so we really think that we're at a point that this can happen, and we're going to implement it. So with that sort of introduction, I'm going to just overview. I'll talk a little bit, an introduction of RNA therapeutics and antisense oligonucleotides, which is a specific type of RNA that we spend a lot of time working on. And I'll talk about two programs that we've been working on for many years now, Usher syndrome and CLN3 Batten disease. So RNA therapeutics just really broadly categorized into these different areas: mRNA editing, aptamers, ribozymes, and oligonucleotides, many different forms of these. And this is really oversimplifying this whole thing. But when we think about RNA therapeutics, the success has been really seen in recent years. And so I brought these out into the classes of marketed RNA therapeutics, ones that have been approved by the FDA or EU organizations. And so we have splice-switching, GAPMers, or RNase H-mediated degradation, siRNAs, aptamers, and mRNAs under these categories. You can see that the FDA approved, we're really getting to have a lot of medicines that are based on these different types of RNA therapeutics. And what's really remarkable is that most of these have been approved since 2016, so in the last 7 years. And for siRNAs, all 5 of these have come since 2018, so in the last 5 years. And so this has been, and now we're seeing clinical trials of these just exploding. And so we expect that there's going to be a continuation of this success in getting FDA-marketed RNA therapeutics. And where has this success come from? Why is it happening now so quickly? And a lot of this is around development. Modifications at of the nucleic acid. So an RNA or an oligonucleotide in its native form isn't a very good drug, but modifications to different chemical modifications can certainly make them more drug-like. These are just some examples that have been developed, and this again is a very small section of this area. It's been a lot of interest in developing even better oligos that have higher specificity and higher efficacy. Delivery has also been a big hurdle for some of the types of RNA therapeutics, and we're starting to see success and breakthroughs there that have really enabled some of these drugs putting conjugates onto the oligonucleotides and delivery through lipid nanoparticles and other nanopolymers. So this has really been key to development and what is really launching this and making this more available. Of course, the pandemic, for all of its bad things that happened. One really exciting thing happened is that we had an mRNA-based vaccine in a year. And so this really brought the attention and the focus on really what can be done with this type of therapeutic. And in the same way, other types of antisense, other types of RNA therapeutics, including antisense, have also followed that path with drugs for very personalized therapeutics, genetic diseases that are very rare, that really going from idea to injection in 10 months. In the case of Melicin, and other examples at the academic level where academic institutions and labs are doing this very rapid development for very specific mutations. And companies and foundations are also being founded around this paradigm. So that has really helped. And so this really has led to the big future of RNA therapeutics, that the market is expected to grow rapidly. We're showing here that mRNA therapeutics market prediction, and it's the antisense and RNAi therapeutics market, where it's really expected to go. And this is really, I think, a pretty, if not a dramatic estimate, but maybe even an underestimate, because I think the potential for RNA therapeutics is vast and enormous. So now let's talk. I want to spend the rest of my time focusing a little bit more on, or actually completely on, RNA therapeutics that function through antisense and complementary base pairing mechanisms. So splice-switching, antisense oligos, gapmers, and siRNAs. With really, I focus on splice-switching, but I want to just call out this mechanism specifically. And so just to go really simplistic, so antisense oligonucleotides work by antisense or complementary base pairing to a target, and in this way they can be directed in a very specific manner to wherever you want them to go into the cell. And the idea is that you can hybridize them to RNA through base pairing and modify gene expression in some way. So there's two types really of mechanisms of action. One we call the occupancy-only mechanism, and one is the RNA degradation mechanism. So let's start with the RNA degradation mechanism. This is really these types of antisense oligonucleotides that bind to an RNA and degrade it through various mechanisms. And this is really based on the chemistry of the antisense oligonucleotide. So RNase H1 targeting to an mRNA through a DNA portion of the oligo will degrade the messenger RNA. They're also called gapmers. And siRNAs work also to degrade the RNA through another mechanism, through antisense oligonucleotides. Now, occupancy-only mechanisms are where the oligo binds and doesn't degrade the RNA. It merely blocks proteins from binding to the RNA or secondary structures of the RNA, depending on where you target it. So you can have an effect of inhibiting translation by blocking the ribosome in some way. You can block microRNAs from binding to the RNA. You can actually activate translation through interfering with upstream open reading frames. A lot of different mechanisms. You can imagine almost any point in the path of gene expression you might be able to modulate up or down regulating by blocking protein interactions that affect the expression. And as I said, we work a lot in the splice-switching ASOs, which is what I'm going to spend the rest of my time talking about, manipulating splicing to recover messenger RNA. So splicing is an integral part in the expression of a gene. Most genes have to be spliced, or the introns need to be spliced out. So in the orange here, I'm representing introns or non-coding portions of the RNA, and then they're spliced out into a messenger RNA that is then exported and translated. So the first story I want to tell you about from our lab is on an older story, but I like to start out with it because I think it shows in a very dramatic fashion the power that antisense oligonucleotides can have for treatment of a disease. And this is Usher syndrome. First I always have to remember to thank the people in the lab that did the work, and this was really a long and very 10 years or so of working with a group of really fantastic scientists that are all mentioned, shown here. And then Fran, Anthony, and Fred, who were in my lab doing the work. So Usher is the leading genetic cause of combined deaf blindness. In its most severe form, there is sensorineural hearing loss. So children are born without hearing, and then they develop retinitis pigmentosa and are almost completely blind, usually in their early teens. It's also accompanied by vestibular areflexia or very severe balance problems. In its most profound severe form, we worked on one of the most severe forms called Usher syndrome type 1C. There's a lot of genes that, when mutated, can cause Usher syndrome. Type 1C is one of the most severe types. And we were working on a particular mutation called USH216G-A. So the USH1C encodes a protein called harmonin, and 216G-A is a mutation, sometimes called the Acadian mutation because it happens primarily in an Acadian population in southern Louisiana, and also in Quebec and France primarily, although it's now being found more frequently in the population throughout the world. So USH1C is shown here, encodes harmonin, and this particular mutation creates a new splice site. So it makes the splicing machinery think that the intron should start here within exon 3, and then when it splices like that, it causes an open reading frame shift and introduces a premature termination codon due to that open reading frame shift. And so you get a very truncated protein and an unstable RNA, and you have Usher syndrome. And so the oligonucleotide strategy that we designed was very simple. We just designed an oligo that base paired across that mutation. And so that splicing site, the splicing machinery no longer recognizes it because the splicing machinery needs to see as an RNA, not. And so this kind of hides that sequence from the splicing machinery. And since you have a perfectly good splice site there, it just redirects it. So it's very simple idea. And a lot of mutations are splicing mutations like this, but very rare ones. And again, so this, usually it's the rare genetic mutations, but they are found. So what we did, we had a mouse model of with the same, where we knocked in the human sequence into the mouse gene. So we were able to make a antisense oligonucleotide to the mutation. And when we injected this, we did a lot of work before this, but I'm just going to cut to the chase here. We injected the mice, and we knew we had to get them into the inner ear, into the cochlea where the hearing, which is the hearing organ and the vestibular organ. We weren't really sure how to do this, but we just did an intraperitoneal injection when they were first born because we know the animals have severe hearing loss or never actually develop hearing. So I just want to show you our mice. And this down in the bottom is a Usher mouse. They run in circles most of the time because they have very severe vestibular problems. And then I will show you when we injected. So here on the right, lower right, is an Usher mouse that we injected with our antisense oligonucleotide when it was first born. And you can see that it's really indistinguishable from non-disease control mice on the top here. And so this was a really exciting result for us, very simple experiment where we just treated the mice when they were first born and a single time, and we were able to completely rescue this circling deficit. We went on to follow these mice for up to a year. Here I'm showing data for 6 months, showing that we really, the mice that we treated never develop any vestibular problems. And we've carried this out now for the life of the mice, and they're really cured of that vestibular deficit. When we look at...
were you surprised by that, that a single dose could have such a prolonged effect?
Yeah, I can't tell you. I think it was probably one of the most surprising things I've ever seen as a scientist. It was kind of in the early days of the antisense oligonucleotide. So again, we weren't expecting much when we did that first experiment, and it literally was those very first mice that we followed out to a year or more, and never had any problems. And since we've done a lot of experiments with the vestibular dysfunction, and we found that we can go down to very, very low levels of oligo. We can treat them out to 15 days and still rescue that, but then we then that goes away. So they need to be treated early. But we think that the vestibular issues are a very good target for this type of antisense oligonucleotide, and delivering in a peripheral manner, not directly to the ear.
Right. So to somewhat follow up on that, you talked about the vestibular issues, but these usher mice also lose sight. Is that correct? Does this affect their sight at all?
Yeah. So the usher mice, we're actually just submitted a paper on that finishes up sort of the first look at the visual deficit. So the mice don't have really the same severe visual deficit that humans have, but they do have visual deficits. And so we were able to show that if we inject, but we have to do an intravitreal injection. So we get a little bit of ASO to the eye when we do a peripheral injection like that. But we had to do an intravitreal injection to get enough there to have an effect on vision. And we also have to, for Usher syndrome, we had to treat the mice every 3 months by intravitreal injections. We've since moved on to do some other diseases, in particular CLN3 Batten, where I'll talk about treating the eye. And for some targets, it seems like the oligo lasts much longer than that. So it seems to be very specific to the target.
Follow-up question on that. Are this ASO in the lipid nanoparticle, or they're just in the solution and when injected?
Thanks for asking that question. I forgot. All the oligos that we work on are naked, so they're taken up. We just inject a naked, modified, and these are all the 2' MOE modifications with the phosphorothioate backbone, and then injected as a naked oligo. So they're not packaged in any way. I should say also, I didn't have this in here, but we've shown that the vestibular dysfunction we can also rescue by treating the amniotic cavity. So just injecting into the mouse amniotic cavity, and the mice that are born don't have vestibular deficits.
Interesting.
Yeah, so that's kind of another interesting thing that we found with the vestibular function anyway. This is just a graph here showing the ABRs, or the auditory brainstem response, measures hearing. And again, we can show the red line is the mice, they don't have any hearing. The blue line is our mice treated with ASO at different frequencies. We measure the hearing. And you can see that at some low frequencies, we get 100% rescue of hearing out to 6 months, although they start to have some hearing loss, so we assume we'll have to do repeat dosing there. But it had a pretty profound effect on the hearing as well. We did a lot more work with this. I talked to you about the vision, the long-lasting effects of the oligos, and the delivery of the amniotic cavity, which I also talked about. One thing that we also did, which I also found very interesting, is we kind of went head-to-head with AAV-mediated gene delivery. So our collaborators, Gwen Jeliak at Harvard, had done some work developing an AAV vector and did the delivery of the hormone. And we found that the oligo performed really comparable to the AAV. And so that was, and for the length of time that they measured out, also out to 6 months. So we learned a lot from the Usher Antisense program, and we're still learning. The vision paper hopefully will be out soon. So with that, we were really excited moving on to some other problems. And we've been working with splice-switching oligos for CLN3 Batten disease now for several years, and I want to tell you a little bit about that. Jacinta postdoc and Matt Stratner, the graduate student, have done most of the work on this, and we work with Jill Weimer at Sanford Research and her group there, along with Dave Pearson and others on this program. We'll mention them along the way. CLN3 Batten is a lysosomal storage disease, autosomal recessive onset. So kids develop pretty normally, and then around 4 to 8 years of age, are usually diagnosed in the doctor's when they go to visit the eye doctor, because they've started to have vision problems, and then usually they're told that something is wrong, and then they get diagnosed. Seizures, loss of motor function, cognitive decline starts around that age, and then premature death usually in the late teens to early 20s. There is some variability in the severity. And there's no cure or effective treatment. It's a lysosomal storage disease, and the hallmark is accumulation of subunit C of mitochondrial ATP synthase and lysosomes. It's not clear why that happens. It's caused by mutations in CLN3. It has a number of names, so that's why this slide has also JNCL, which is another name for juvenile neurosteroid lipofuscinosis. Most cases are caused by a deletion of exon 7 and 8. And so here is the CLN3 gene, 15 exons, some lysosomal targeting sequences. It's a membrane protein in the lysosomal membrane. We don't know what it does. The most common mutation, deletion of exon 7 and 8, 80% of patients. And so this results in a premature termination codon exon 9 and a loss of function. We've shown that that's an unstable RNA through nonsense-mediated mRNA decay. Some people believe it could also have a gain of toxic function. There's, when we started to think about how approaches that we could take to treating this disease, one thing we noticed about it is that it's highly alternatively spliced. So there's a lot of alternatively spliced isoforms. This is showing just some of the ones that have been annotated in Ensemble. And this green orients you to the fact that one of these isoforms is skipping exon 5 as well as exon 7 and 8. And this is in a normal sample. And so one idea was, well, maybe we can shift the splicing towards a naturally occurring isoform. And the thing about this naturally occurring isoform is if you skip out exon 5 from the transcript, you restore the open reading frame. So you no longer have a premature termination codon. You still have these lysosomal targeting sequences. We predict, fewer transmembrane domains in the membrane, but our hypothesis was that this could rescue some of the function that was lost by the mutation. And another thing that sort of supported this rationale is that some mutations in this gene, mostly missense mutations, cause a much less severe disease, just isolated retinal degeneration. So our idea is we could go from a severe, deadly neurodegenerative disease, correct the open reading frame, and get something that is much less severe. And so that's what we did. Again, we were using these modified oligonucleotides of phosphorothioate backbone with the 2' methoxyethyl modification, and also just naked oligos. We did a lot of screening across the introns and exons to identify the most active oligonucleotide. This is in vitro. And then we had 2 mouse models that we were working in. One was a disease model where delta 78 was knocked out, just like in the humans. And then the other one is actually a therapeutic model where we asked the question, if the mouse is just expressing this isoform, the lacking exon 5, 7, and 8, similar to what we would be doing with our ASO, is that mouse okay? So those were the 2 mouse models that we wanted to really show that this would be a good approach. Unfortunately, the mouse model does not have a severe disease like humans, which is not uncommon for mouse models. It has deficits in motor skills by 8 to 12 weeks, and it does accumulate out of this storage material that's a hallmark. So we went about first injecting the oligonucleotides very early into neonatal mice. We know that the storage material starts to accumulate very early, even embryonically, so we wanted to go in early. We treated, and then we looked some behavior assays and did analysis of splicing. The first thing we found we're really encouraged by is that the oligonucleotides after one treatment by ICV injection at P1 lasts out to 14 months. At 14 months, it still has about 50% of its activity. Before that, it's pretty active oligonucleotide, pretty stable activity. So and we found this throughout all regions in the brain. So it's widely distributed, around 60 to even up to 80% skipping throughout the brain. I don't want to spend a ton of time on this, a busy slide, but we did show rescue of a decrease in the storage material in the different. So blue is our treated mice, red is our mutant mice. So a decrease in the storage material across different brain regions. And the mouse phenotype is how long they can stay on a accelerating rotor rod, and also some motor coordination deficits in turning around on a vertical pole. And we found that these activities were rescued in our treated mice. So that was really encouraging to think that then we could recover some phenotypes in these mice. We created a severe mouse model where the mice actually do die, have a premature death at about 18 and a half days is 50% survival. And we found that when we treated those mice with our exon 5 ASO, we were able to recover survival out to the level of a heterozygote mice in this severe model. And so again, this also is showing in a severe case that we can have some benefit from skipping this exon in CLN3. So we were really excited about that. And I told you we also have a disease model where we just genetically made the exon 578. So the mouse is only making this CLN3 with lacking exon 5 and 7 and 8, so only making that isoform. I'm not going to get into that paper. We also did similar showed behavior rescue in the rotor rod and in a string pull test, which shows fine motor coordination. And we just recently published this and showing this result. And so again, supporting sort of further development of this for the treatment. And so I actually just want to end really quickly on one final comment, sort of on a hope that we have. So I told you that about 80% of the people with Batten disease have this delta 7/8 mutation. But what we've been seeing in the community is a lot of development or pursuing of these treatments using ASOs for rare disease. FDA issued guidelines for the use of antisense oligonucleotide therapeutics for personalized therapeutics for N-of-1 type of trials. And this series of guidelines has really helped researchers such as myself to think about treating not just those rare diseases with that are 80% of a mutation, but the rare ones also. And if you look at CLN3 Batten disease, for example, even though 80% of the population have deletions of exon 7 and 8, there are a lot of other disease-associated mutations throughout the gene. There are missense mutations that I said most of these are milder forms of the disease, but there's a lot of loss of function termination and splicing mutations that we think with this guidelines from the FDA to do personalized N-of-1 that it's possible to develop very quickly a potential therapeutic for these rare cases. And so in these cases of N of few, can we then take and target splicing in some way to get a functional CLN3 protein isoform? And so those are some of the things that we're working on and some of the things that we hope to bring as we go forward and start thinking about for other labs, along with, of course, treating the bigger problems as well. And with that, I am going to stop and just thank the people currently in my lab and the former members that have been instrumental, of course, in doing all this work, and the collaborators. Many of them I've mentioned along the way, and the funding sources as well.
That was spectacular. Thank you so much, Professor Hastings. I'm going to let my junior colleagues ask a few questions, but I do have something for you in just a moment. Go ahead.
Thanks, John.
That was great, Michelle. So I'm kind of curious how you choose the disease that you're going to work on. Like what will be next your criterion for assessing what's amenable to splice-switching oligonucleotide therapies? What's the process there, I guess?
Yeah, I know. It's a good question. So it's changing a lot now as with these guidelines. So it used to be really that having a good assay for it, having animal models, things like that, would really help get things started. And also having a good rationalized target. So the Usher syndrome was an easy one because the splice switching, the cryptic splice site that was activated, it was pretty easy to convince people, like if you block that and you're just going to make the normal protein again, right? And there was an animal model, and so that was sort of an easy one. The CLN3 batten we worked on, even though it was an uphill battle convincing people that that was going to work or have any sort of effect. I feel very convinced now that it is, and when we've had other people working on this, other labs that have started working on this, applying their assays and showing that it also does seem to have some function. So, but, again, there's so many easy ones to potentially do, like the Usher one, but they're so rare, right? And so those you have to ask yourself, is it worth doing? But I think now we're seeing that the future is coming, where we'll be able to design these very easily for a cryptic splice site and an intron, that we can just design it, show it's safe, and have effectiveness through, and really on the hinges on the heels of all of these examples that we're seeing in all of these, of course, not just my lab, but many labs that are working on this for different diseases and different tissues.
Right. So I guess I'll jump in with another question. The approaches that you took to go after the Usher mutation and the CLN3 batten mutation were dramatically different, right? You were able to just revert splicing to the normal place. And for the CLN3 mutation, you didn't try that. Was that because there were just too many individual mutations for you to go after that 80% as a group?
For the CLN3, hopefully I understood this question right, but because these types of deletions are so devastating, right? There's just not any good way to correct them other than maybe gene therapy, RNA therapeutics, right? When you have a large deletion in a gene, right? So that's difficult. Gene therapy approaches for CLN3 batten have not been successful yet. I was working with groups that are doing that, and that we're really hopeful about it. It just seems like this gene is very tightly regulated, and it's turned on in we don't know where, but it seems to be highly regulated at the protein level. We don't actually have a good antibody either. So I guess what I'm trying to say is that even those approaches where mRNA or gene therapy, they're not very useful because you don't know where you need to express it and when you need to express it. With ASOs, at least you're targeting the endogenous gene, so you take that off the table. And so that's another thing that we think about when we think about using ASOs versus RNA therapeutics or other things is how much do we know about it? Is it regulated? How are we going to be able to turn it on the right place at the right time, the right level kind of things? Or do we need to rely on an endogenous targeting where, if that's the case, then a deletion, you just kind of got to do what you can.
Right. And I guess another question along that line is, there's always the chance with a gene therapy or an mRNA therapy that overexpression can cause its own problems as well. Would you have any data to suggest that one way or the other?
That overexpression with CLN3? Yeah, I mean, we have, because we don't have a good antibody, that a lot of studies have been done on the overexpressing, and it doesn't seem like overexpressing wild type has ill effects. It does seem like that overexpressing the mutant has ill effects, and so that's another caveat, that if there is a toxic gain of function element to this disease, doing a gene therapy or an mRNA is not going to necessarily be helpful, at least in the case of a splice-switching oligo. You're going to take care of that too, because you're going to be getting rid of any dominant negative effect as well. And so we definitely need to learn more about this disease, and a lot of disease, some are easier than others, but this one, but we'd like to advance something before we know everything. I think we're getting to the point where we don't necessarily need to know everything.
Okay, I have a question regarding the delivery mechanism in human. And I know the splice-switching oligos can be very expensive. So what do you think about bringing the cost down for the ASO, and what would be the best delivery mechanism you can think of?
Yeah, I mean, I think that's a good question. I mean, the cost, is high now of making them even in the lab, right? And so that's one reason we are starting this RNA initiative here, is to try to synthesize in-house, so that maybe will help with the cost and make them more available to the basic researcher. But you're right, in terms of treating humans, I mean, the central nervous system is one target organ that we like to work in, because it requires lower doses than the rest of the body, and the oligos at least are more stable, seem to be more stable there. So they get into postmitotic cells, so you don't have to dose as frequently as you might have to in the periphery. But certainly in the periphery, trying to target it to specific organs is a really important goal. They have things like the GalNAcs I mentioned that are helping get them delivered to the liver, specific cells. But I think that's a really exciting area of research, trying to put ligands or conjugates onto these oligos to target them specifically to the tissues or cells that need to be expressing them.
I have a question. It's part of a curious comment question. How do you think, take into account the specificity of based on their size? They're relatively small. How do you is there a screening process that you use to select whether what's going to be the average or the appropriate size of that ASO in order to be specific to that target region that you are selecting?
Yeah, I mean, that's a good question. That's a bit important question. There's a lot of talk about that and a lot of investigating into that. So the first thing you want to do is to find the best ASO, the one that's the most active. But of course, you don't want it to have a lot of other hybridization sites in the genome or on the RNA. And so we usually typically will go for the best one, and then we will. And then we search for potential off targets. So we usually do that through just looking for things that are targets in the genome, and again, looking really for expressed RNA. With the steric blocking ones, we feel that the landscape is a little more forgiving, because you have to really block a protein binding to probably have any effect. But we usually go and look. So a lot of people also look for off-target by doing sequencing, RNA sequencing. This is it's a difficult, I find that to be difficult, because of course, if you're doing that in a disease cell, you might be having a lot of effects that are nothing to do at all with so that can be problematic. And then of course, there's also some evidence in some cases where having a mismatch might actually improve the activity. Adrian Krainer's lab came up with something like that too. So it's something that it's a concern. It's with the splice switching, because you're not actually, I feel like, cutting your DNA or doing something that it might be more tolerated. I mean, small molecule drugs that we use every single day have side effects and off-target effects, of course. So that's not necessarily a great answer. There's definitely need to try to try to minimize that.
In the time we have remaining, Michelle, I have two questions for you to consider. The first one is, tell us what hurdles you see going forward for your program in the regulatory roadmap to the clinic. What are the hurdles that you have to overcome in your program? And then finally, maybe to end, you could tell us a little bit about the Society for RNA Therapeutics and your role in it.
Oh, okay, great. Yeah, so I guess in terms of actually getting really to the clinic, of course, with rare disease like the CLN3 bound, the 80%, this is still a rare disease, right? And yet it's still too many kids with the disease to be able to do an N of 1 trial, sort of at a small scale where you don't have to do a full-blown clinical trial. And so that's still something that we struggle with, is how to convince industry partners and how to develop, the cost that it takes to do a clinical trial. So that's a practical thing. I think the efficacy of the for the CM3 batten in particular, showing the function of the protein and things like that is something that we are definitely working hard on with biomarkers and things like that. With the NF1, again, this is something that academic labs are doing through clinical partners, and so I find that really an exciting way to reach more people and that can be helped, because now I think we are at a time, this sort of a frustrating time where we know for some patients with disease, we have the ability to help them. We have the ability to help them. And it's just a matter of making it happen. And so that's something that I really am passionate about getting through and finding a way to help them, even though, right, we're at the beginning, right? We have our FDA guidelines, people are starting to do it. We're going to have to work on the kinks, obviously. I'm really excited about the Society for RNA Therapeutics, because this is, again, part of the process and part of this infrastructure and the education and the outreach and the partnering that we need to get everybody sort of talking about it, doing it right, collaborating, working, learning from each other's mistakes and victories, working not like in a competitive manner, but in a cooperative manner, right? I mean, there's so many people, there's so many rare diseases and so many mutations. There's definitely no room for competition. There's just a huge unmet need.
Well, it's wonderful to have you on the show today. I'm excited to see your work going forward. You're really one of the pioneers at the cutting edge of RNA therapeutics, and such a pleasure to see you today, Michelle, and then hope to work with you more in the future. Well, you've been at the cutting edge of RNA therapeutics today. Our guest today was Professor Michelle Hastings from University of Michigan, telling us about splice-switching oligos for genetic diseases. Thanks again, Professor Hastings, for joining us today.
Thank you so much. It was my pleasure.